Cool. So, first of all, you know that in your work that you deal with a section of human reproduction. And what is very important for you to remember is that you relate that work to the work that we did yesterday. We undergo sexual reproduction as a human being, so do our angiosperms, so do the birds and the bees and whatever else it is, so that we can give rise to genetic variability within a population. Remember, what is very important is that every organism in a species must be different. And the reason why they must be different is because that genetic variability is what guides natural selection, and that natural selection is, of course, what guides evolution. So you need to bear in mind why there is genetic variability in the first place. Number one, when we undergo meiosis, you know that in prophase 1, we did this yesterday, there was crossing over or chiasmata that occurred, where the homologous pairs sit together and maternal and paternal genes swap between each other, creating genetic variation in the chromosomes. We know that in metaphase 1, when they line up along the equator in their homologous pairs, they line up randomly, which means there is always a different combination of maternal and paternal genes that are lined along the equator. That also gives rise to genetic variability. So therefore we know that every single sperm that is made, every single egg that is made, is always going to be different from any other one that was made. As I say, genetic variability. Also remember that why do we undergo sexual reproduction? Genetic variability comes about because we combine two different gene pools. We know that in meiosis we make haploid gametes, haploid sperm and haploid eggs. In other words, in a human being, instead of having 46 chromosomes, like in a normal body cell, we have 23 in the sperm, 23 in the egg, so that two different gene pools can come together in the sperm and the egg and come together in fertilization, create a zygote, and that zygote then grows into the new individual. Very important that you always bear in mind sexual reproduction is about genetic variability. So the first topic we want to start looking at here are aspects regarding the male. And the first question is study the diagram below and answer the questions that follow. So we have a diagram over here, and please remember, learners, always look at your resource material, familiarize yourself with the resource material before you start looking at the questions. So we can see this is the human male reproductive system, and of course the human sperm cell. So let's now start on looking at our questions. So it says provide labels for A, B, E, and G. Remember that even though this is just A, B, E, and G, 1 mark each, remember I'm teaching around these concepts. So you as a student would just write the particular label down, but what I'm going to do is not only give you what the label is, but also tell you obviously what is going on in that part of the male reproductive system. So the first of all, it says label A. So if we look at A, we see that it takes us to the prostate gland over there. So A is our prostate gland. Remember that when the male ejaculates, he releases semen, and sperm are inside that semen. And the main gland that is involved in the production of semen is, of course, the prostate gland. So that is, of course, our A. If we look at the next one, it says provide labels for A and B. There is B. I see B goes to my sperm duct over there, which is also called the vas deferens. Now, what is the purpose of that B? Remember that when the male is releasing his sperm out of the body, or when he is ejaculating, we know that seminal fluid is produced by the glands over here, the prostate gland, the cowpus gland, and the seminal vesicles in this region over here. But the sperm that are made by the testes must pass up the sperm duct, collectively join with the semen that is produced by these glands, and then it'll be ejaculated out of the urethra. Obviously, the man would have an erection at the time, which is not shown in this particular diagram over here. So that is what our B is. E, if we look at E, E is the urethra. So what is important about the urethra? The urethra in the male, which is different to the female, is that the male releases his semen and sperm through the urethra, and he releases his urine from the bladder, down the urethra, through the penis, and of course out of the body. And finally, it says, what is G? Now G brings us to this diagram over here, which is of course the human sperm cell. And I can see that G is inside over there, the head of the sperm, and therefore that is the nucleus. So remember, the only thing the male has to deliver in the process of human reproduction is to deliver his genes, his 23 chromosomes, to the egg for fertilization. That is found in the head, and that is in that region there, G. You learning something, Amanda?
I'm learning something. 23 chromosomes. Okay.
Jolly good. Excellent. Right, if we look at our next question, it says, state one function each of C and F respectively. Right, so let's look first of all, see what C is. C is this coiled tubule on the outside of the testes. It is a structure that is called the epididymis. Now remember that when the male ejaculates, he does not release sperm from the testis itself, he releases sperm from the epididymis. So sperm that are produced in the testes go into the epididymis over there, our C, where they will undergo their final maturation, number one, and of course they are stored there so that when the male releases his sperm, these sperm will travel from the epididymis up our vas deferens and of course out of the body. The second one says state the function of F respectively. Now F is the structure over here at the top of the sperm. That is our acrosome. And what is the purpose of the acrosome? It contains enzymes. So that when sperm come along, we often see that there's an egg and lots of sperm are sort of banging their heads in there to try and get in. One sperm's acrosome opens, it dissolves a little hole in the membrane of the egg, and this head of the sperm gets in. Remember, the only thing that the man leaves is, of course, the head of the sperm inside the egg, and therefore it has undergone fertilization. These two structures, the middle piece and the tail, are there purely to propel the sperm towards the egg.
So it helps it in terms of speed and stuff?
Yes, it just moves it along up the from the top of the vagina, through the uterus, into the fallopian tube.
Oh, okay.
Okay, it's very simple for us males. We've just got to deliver some sperm, and the rest is all up to you. So if we look at question 2.1.3, it says state the letter and the name of the part where sperm are produced. If I come back to my diagram, that is over there, the testes. So the letter is D, and the testes is, of course, the place where sperm are produced by the process, remember, of spermatogenesis. If we look at question 2.1.4, explain why it is necessary for part D to hang outside of the body of the male. Now, of course, our part D is the testes. Now, we see that it hangs out of the abdominal cavity. Now, the reason why the testes must hang out of the abdominal cavity is that our normal body temperature is 37 degrees. Sperm do not form at 37 degrees. They like to form at about 34, 35 degrees. And therefore, by keeping the testes in the scrotum out of the body, we therefore ensure that the right temperature is maintained and that sperm will be produced. Okay, I think we've got time for one more before the ad break. Name the following: the cells that secrete the male sex hormone. They are inside the testes. They are called the interstitial cells. And the hormone that stimulates the development of secondary sexual characteristics formed or produced by these interstitial cells is, of course, the hormone testosterone. I think we are moving into a